Welcome. Welcome to the SeaWorld Research and Rescue Center. I am Dr. Elise, and I thank you all for attending this new science talk of the 2025 series. As you know, this series of science talk at the center is developed in close collaboration with the Environment Agency Abu Dhabi, the Emirates Natural History Group Abu Dhabi, the Natural History Museum Abu Dhabi, and Nautica Environmental Associates. So I thank you. All of them are represented tonight, so thank you everyone for being here and for supporting us. For tonight's science talk, we are delighted to welcome two visiting researchers from James Cook University, all the way from Australia. If you have any interest in dugong ecology and conservation, you may already be familiar with the name Professor Helen March. And if we can have some picture, oh yes. So Helen is a world-renowned expert in dugong research. She has authored numerous publications and a seminal reference book on the subject. Also, Professor March is not with us today. She knows Abu Dhabi very well. She's been working with Dr. Das for many years and collaborating with the Environment Agency Abu Dhabi. In fact, she visited us just a few months ago. Those pictures are from that, and she did spend some quality time with Malkout. This ongoing collaboration has led to this week's visit by René Lambourne and Sarah Landeo-Yori in partnership with the ED. René and Sarah are both members of the Dugong research team at JCU's Center for Tropical Water and Aquatic Ecosystem Research, TropWater, yes, led by Professor March and Dr. Christopher Kleger. TropWater conducts research on tropical aquatic ecosystem across northern Australia and beyond, with a focus on sustainable management and global ecological understanding. Tonight, we will hear directly from René and Sarah about their PhD research on dugong ecology, two fascinating studies from TropWater. We'll also find out what inspired them to travel all the way from Australia to the UAE and to meet Malkout. Before I start introducing our speakers, please allow me to remind you to turn off your phone, and I hope you had enough food, enough water. Please refrain from going in and out of the auditorium as much as possible. Thank you. All right, we'll start. And we'll start with Sarah. Sarah Landeo-Yori is a PhD candidate at James Cook University in Australia. Her research focuses on developing a non-invasive method to assess the body condition of dugongs using drone videos and images. Sarah has previously worked with other Sirenian species like Amazonian manatees in Peru and Antillean manatees in Mexico. She is very excited to be working with dugongs now, and she will be telling all about it. Come on stage.
Thank you for having me today. Thank you, Dr. Elise, for the presentation. Tonight, I'm going to tell you a bit about my thesis project, which is basically using drones to assess dugong body condition. But actually, I'm going to talk about how to measure a dugong without touching it, and how is this related to using drones to assess or monitor dugong health? I would like to bring attention first to health, animal health, and why it is important. The main reason is health is directly linked to survival. So the health status of an individual might affect his capacity to migrate, to defend a territory, to acquire food, or to for mating processes or breeding processes. Populations that are healthier have a better capacity to withstand environmental pressures, and also the health of the populations. It's an indicator of how healthy is the ecosystem as well. So in sum, to be able to do a correct management and conservation of wildlife populations, it is important to consider the health factor. Well, here we have a group of dugongs in Moreton Bay in Australia, as an example of several individuals of a population. And just a reminder that it's important to assess the health of these wildlife populations for management and conservation. So let's talk more about dugongs, which is the focal species of this study, my study. So dugongs are marine mammals. It's the only species of the order Sirenia, which also includes manatees, that is exclusively restricted to marine environments. Dugongs eat seagrass. They are seagrass specialists, and therefore highly dependent on these habitats. Also, dugongs and manatees have populations that grow very slowly, because, for example, the gestation period is about a year, 14 months, and the period that the calf is dependent on the mother is about 2 years. So let's say that every 3 years, a mom can have one calf. So populations grow very, very slowly. Just in a general way, you can see that that's the distribution range of the species, the dugong, from East Africa on the west to Vanuatu in the east. It spans across 37 countries, and the biggest populations can be found in Australia and throughout the Arabian Gulf. So the threats to dugongs also might vary between different populations, but in general, one of the main threats has to do with their habitats, with seagrass ecosystems, because seagrass ecosystems are very fragile and they can be affected by climate change and coastal development. Other direct threats include vessel strikes, entanglement on fishing gear, and more directly, hunting practices that are non-sustainable. So in general, this species is considered as more vulnerable, but in some regions, it's locally extinct or endangered. Okay, but going back to health. Well, we know that health has many components. We can assess health using a blood test, maybe an ultrasound. We could be looking at the parasite load, but this project has to do with nutritional health, and more specifically with body condition, which is an indicator of nutritional health. So here in the image, you can see 3 different manatees with 3 different body conditions, and just looking at them is like, one is fatter than the other, there is one that is more slimmer. So basically my project has to do with assessing how fat are the animals, how good is their body condition. Because this reflects a balance between the energy intake and the energy expenditure. In other words, they're fat reserves, right? The energy bank that they have and need to use for many, many processes. So what factors affect dugong body condition? Let's imagine that energy is like currency and the body is like a piggy bank. So how do we obtain more energy? Basically, food, right? We need to eat. It's the same with dugongs. But with energy expenditure, the budget can be very large, from basic metabolism, movement, thermoregulation. If it's a juvenile or a calf, they need to grow. If it's an adult, they need to use energy for mating, for pregnancy, for lactation, to maintain their immune system. So it's a very, very long list. And also, the factors that affect the dugong body condition might change across populations, depending on the location and the environments. For example, seasonality might influence the seagrass availability and quality. And also, in terms of energy expenditure, seasonality, if it affects the water temperature, for example, let's imagine that during winter the water temperature drops, the animals will need to spend more energy for thermoregulation. We also have other local factors, for example, human disturbance. They might increase stress levels, therefore more energy might need to be used to maintain their immune system, predation, competition, seagrass dieback events, etc. So in general, it's important to understand how body condition might change across populations. Considering temporal scales, for example, through the year, the importance of the seasonality, but also spatial scales. And what can we learn from this? Well, we could have a baseline for monitoring and conservation strategies if we assess the health of a dugong population this year. This is the baseline for the monitoring of next years. Is this changing, is increasing, is decreasing? How resilient is the population at the moment? We could also detect some warning signs about the ecosystem decline if the population health is not good. And we can also learn about bioenergetic dynamic. For example, migration costs. How is the body condition before migration? What is the body condition after migration? But how do we assess body condition, particularly dugong body condition? Well, we use body condition indices. And these body condition indices are typically obtained using the relationship of several 2 or 3 body measurements. For example, body length and girth. And here we have an example. This body condition index, for example, uses the umbilical girth and is the ratio of the umbilical girth and the body length. It can be also the residuals of the regression of these variables. But maybe let's look at something that might be more familiar for us. This is body mass index. We humans also use this type of body condition indices to assess if we are in a normal weight for our size or if we are underweight or overweight. So this is also using body measurements like weight and height. In the case of dugongs, how is this morphometric data obtained? Well, as humans can just measure ourselves or use a scale, but for dugongs, maybe, or in general, wildlife is more complicated. There is a lot of information that can be obtained during captures that are part of generally long-term health assessment programs. And there is one in Australia, actually. I think it has been running for probably more than 10 years. But even though we get a lot of information from these captures, it's not only body measurements, we can get information on blood or on hormones. This can be also very expensive. These captures require high budgets, complex logistics, a lot of training, and a big team. And it might involve risks for both researchers and the animals. So therefore, to obtain body measurements, maybe there are alternatives that are more cost-efficient and non-invasive. And one of these alternatives is photogrammetry. Photogrammetry is a technique that allows to measure the size and shape of objects from photographs. And it's less expensive and invasive than methods that require the direct handling of the animals. For marine mammals, photogrammetry has been used since the 50s. The easiest way to scale the size of an animal in an image is to use a reference object. If we know the size of the pole, and we can scale to the size of the animal. images for photogrammetry have been obtained from land, from a boat, underwater, with helicopters. And as I was saying, for example, in the image here with the dolphin, I don't know if you can see, but there are 2 dots near the dorsal fin. Those are laser dots, so it also provides a measuring and a scale for scaling. But then drones appeared, and drones have significantly expanded the possibilities for aerial photogrammetry because they can provide high-quality aerial imagery. And they have already been applied to successfully obtain morphometric data to assess body condition of a range of marine mammals, especially cetaceans and pinnipeds. Very little has been done on sirenians. To my knowledge, there is no study for wide free-ranging populations of sirenians. There is one study with manatees under human care. So there is possibilities to explore this, and actually my project has a lot to do with this, to develop a method that can be used for dugongs in particular. So what is the aim of this project? Basically to develop a method for assessing dugong nutritional health, evaluating body condition, and to apply this method to investigate seasonal and spatial variation. Well, basically what we do is a lot of drone deployments, and we search for dugongs. My main study area, it's in Australia. I will show you a map a bit later, but basically what we do is using drones, and from very high up in the air, we try to look for dugongs. This is a group of dugongs in Cleveland Bay in North Queensland. As you can see, there is a very distinctive, we call them feeding plumes. When the dugongs are feeding on the bottom in very shallow areas, there is a lot of sediment coming up, and the sediment creates these plumes, and they are very easy to detect from the air. So once we have located a group, we focus on 1 or 2 individuals. Well, here are some examples of drone images obtained from the dugong population in Cleveland Bay. As you can see, the waters are a bit turbid there. However, under the right conditions, it's possible to obtain some images. And you might notice that we are actually looking for specific positions, right? Dugongs in straight position, whose body shape is visible, is what we need to be able to obtain the measurements that we need. So basically, as I was already saying, the data collection implies flying a drone, obtaining a very good quality video, and we are also using a LiDAR altimeter, which I will tell you a bit more later, but we are measuring the exact altitude or height of flight for our scaling processes. We are flying generally at 20 meters high, but with some drone models that have optical zooms, we can fly higher and still obtain good images. After we have obtained those videos, the videos are reviewed, and some still frames, the best shots with the right position, are extracted. Then these go through a process of image scoring. Some images are selected, the best one, the one that they need to fit some requirements in terms of straightness of the body, the resolution. But then with these images, it's possible to do the image analysis and the scaling. So this is the part of how to scale pixels to metrics, how to actually measure the animal without touching it. We need to calculate this ground sampling distance. Is basically the size of a pixel, how on the ground, how many centimeters or meters it represents. And for this, we need some information. We need camera parameters, the sensor width, for example, in this example, we need the focal length, Then the flight height, in this case, that is why we are measuring with precision the flight height, and then we know the width of the image footprint. The relationship between these parameters allows us to know what is the ground sampling distance, how many centimeters are in a pixel, each side of the pixel, because it's a little square. But basically, we use this information. So for my project, we are working on developing body condition indices using drone-based photogrammetry data. It's not only one, and we were talking about, for example, length and girth, but we want to test several others, starting from the very simple ones, maximum width and body length. And this is a dorsal image that we can obtain with a drone, for example. But we would also like to test girth versus body length, and surface area, and volume, and potentially mass. So, but let's think again. Drone images can provide overhead view of the animals, and we can get, with this is a whale example. We can get the body length, we can get the width, we can get the surface. This is a dugong, so it's possible to do the same for dugongs. But what about the girth and the volume estimation? So let's look again at this whale example. So we have the dorsal view, but here the whale also has well, we have the lateral view of the whale as well. And with this information, it's possible to model the volume of the body. We have information on the widths along the body, we have information of the depths along the body. We can get, for dugongs, this, but we need this information too if we want to model girths and volumes. So lateral images are needed to obtain the ratios with depth along the body, like several slices, and what is the relationship for each slice, let's say the dugong body shape. And these ratios should be similar across individuals because they all share a dugong body shape. So what can we do? What can we use to obtain this information? So we need morphometric data to validate this, and this data can come from morphometric data and dorsal shot from dugong captures. This is actually one image from René's fieldwork last year, because during the dugong captures we're also measuring them. But also, we can use morphometric data and associated dorsal and lateral shots from dugongs under human care. And this is where Malkut comes in, and how is he helping this project. Because as you can see, well, we can actually get very good lateral images of his body as well. And yeah, so Malkut will help us because he will provide reference information on the ratios with depth along a dugong body. We also have taken dorsal and lateral images, and also manual measurements of the body to adjust the modeling. Additionally, if we have the body weight and the volume, we can calculate the body density and use this as a factor to estimate weights on dugongs in their natural environment. So just an example of what we have been doing these years. This is a dorsal view from Malkut, and this is actually a reference object. We know the size of this little square, so it will help us during the scaling of pixels to metrics. This is a great lateral view of Malkut, and we also got some nice lateral views underwater. We were also working on getting the body measurements, the weight, and yeah, even ultrasound to help us understand how the thickness of the blubber layer varies across the body. And well, I just want to say thank you to the amazing research and rescue team for helping us with this process. So in summary, body condition is an indicator of nutritional health. Drone-based photogrammetry can be used as a non-invasive method to assess dugong body condition. Several body condition indices will be tested as part of the method development, and the estimation of girths, volume, and mass requires reference information like the data provided by Malkut. And at the end, the application of this method should enable the monitoring of nutritional health in dugong populations, which will inform conservation and management programs. Okay, thank you very much. Okay.
Thank you, Sarah. That was absolutely amazing explanation of your study. I'm sure there is a lot of questions to come, but let's hear Rini as well. And I'm sorry, I'm going to pronounce it à la française, René Lambourne. I don't know if it's like this that you pronounce it, but Rini is a PhD candidate at James Cook University as well, and the Harry Butler Institute. Yeah. Reni's focuses on understanding the fine-scale behavioral ecology of dugongs using multi-sensor tags, which record movement, behavior, and the physical characteristics of the animal's environment. So let's welcome Reni on stage.
Yeah, thank you. My name's Renee, and today I'm presenting a little bit of the PhD research that I'm doing at James Cook University. So animal behaviour is underpinned by movement, which is the response to either internal or external stimuli. Therefore, behaviour is at the basis of applied ecology and is important in understanding individual, population, and ecosystem processes. This means that before the potential effects of any threat can be fully understood, it's important to understand the behavioural ecology of an animal. However, historically, the behaviour of species that are elusive or inhabit remote locations, such as marine animals, have been challenging to study. For example, cryptic species of marine megafauna may spend just a few seconds at the surface, and this is the case for dugongs. So movement is driven by the physiological needs of an animal, from nutrition to thermoregulation. In the case of dugongs, diving duration is limited by the need to return to the surface to breathe. So dugongs must divide their time between diving to rest, travel, and for resources like food, and returning to the surface to breathe. Therefore, dive duration and foraging time is limited. So, given this, dugongs must satisfy the energetic requirements for physiological processes, such as metabolism and growth, in a limited time. However, little is known about the diving behaviour of dugongs. So we don't know how dugong diving is associated with environmental factors, or how it varies between habitats, such as between intertidal and subtidal habitat, and by extension, how behaviour may be affected by changes impacting dugong habitat. So we focus on foraging dives. As seagrass community specialists, foraging occurs at the bottom of a dive, where dugongs are at the sea floor. Therefore, foraging time is limited to the proportion of time between the ascent, which is the red line, and the descent, which is the purple line. This is important as any impact that may affect the presence or growth of seagrass will inevitably affect dugongs, as they feed almost exclusively on seagrass. And Sarah touched on this with body condition of the dugongs. So, how would dugongs adjust their diving behaviour to compensate for any change? Well, at the moment we don't know. Will dugongs opt to perform more dives in shallower waters where seagrass may be a bit more scarce? Or will they opt to dive deeper where there may be a higher density of seagrass, but the cost of diving to those deeper depths may be higher? So, we don't yet know how they adjust their diving behaviour. and how they may compensate for any change in the environment. Therefore, investigating the physiology, diving behaviour and activity of dugongs may provide an understanding into the energetic impacts that threats may have. So to date, dugong diving behaviour has primarily been studied in 2 dimensions, so looking at depth versus time. And if you have a look at the plots up on the screen, there's also different shapes of dives. So we have depth along the y-axis and dive duration along the x-axis. However, the diving behaviour of dugongs has mostly been inferred from these shapes. We don't actually have any information on the activity that the dugongs are doing during the dives. So we can't definitively determine the behaviours that they're performing. Other studies have identified 3D space use of dugongs by combining time depth data, which we saw in the previous slide, with GPS points. So they have an idea of how dugongs are using the depth, but because there's no activity used, we still don't know what the dugongs are doing within the space. So we can build on these studies by deploying tags which incorporate accelerometers, and accelerometers measure activity. This will provide information on what the animal's doing and where it's doing it. And when activity is used as a measure for animal movement, we can reveal different functions of dives. So technological advances and the development of animal-borne biologging, or multi-sensor tags, provide an opportunity to gain information on the diving behaviour, physiology and activity of a species. Biologging's been well exploited by researchers that have studied seals and penguins. There's been a lot of research on those, as well as dogs, badgers, all sorts of animals. So from this, they record animal movement, behaviour and energy expenditure. In the form of diving behaviour, we can get stroke rates, which if you have a smartwatch that records steps, that's essentially the marine animal equivalent of steps. Swimming strategies, so some seals, for instance, do swim and glide strategies, so they'll swim for a bit and then glide to conserve energy. We can also identify buoyancy control in marine animals, as well as foraging strategies. Now, I mentioned accelerometers before. Equipping biologging tags with accelerometers has significantly improved behavioural interpretations, as acceleration contains 2 signals. The first is body posture, which we see on the left. And if you ignore the names on the side, they're the 3 acceleration axes. So they correspond to up-down movement, side-to-side movement, and surging, forward-backwards movement. And we can see for a leatherback turtle that in the middle axis, there's a big peak and trough in the data. And that shows that the leatherback turtle's rolling from side to side. Acceleration also reveals dynamic movement, so such as flying, swimming, and running. Now, it's important to include activity when calculating animal time activity budgets, because we can identify a balance between conserving energy and energy expenditure. Because activity and energy expenditure are linked. The more activity, the more energy expenditure, and the more energy is needed, not just for movement, but for growth and reproduction. So this kind of leads me to my project. So multi-sensor tags give us an opportunity to gain insight into the links between dugong activity, movement, and behaviour. And for animals like dugongs, where direct observation of behaviours is difficult, being able to deploy biologging tags provides an opportunity to gain an insight into what they're doing underwater. So for my study, we've been deploying archival mini diary or camera diary tags. Now we've only deployed 1 camera diary. The other 29 have been mini diaries. And actually I'm going to pass one around so you can have a look at what we've been deploying on the dugongs. Now, these tags have a heap of different sensors inside them. The most important ones are depth, acceleration, so it measures in 3 dimensions. It's also got a magnetometer, a gyroscope, and it measures temperature and light. And we program the tags to record 50 times a second. So we get a lot of data. But this is important because when we record at that high frequency, I can see every fluke beat, which is like the tail of the dugong, and I can see every head movement when the dugong's foraging. This should play. It's just a video of a dugong swimming away with a tag. Oh, it's playing again. Now, because the tags are archival, we need to get them back. Otherwise, I don't get any data. So the tags are equipped with a VHF transmitter. And the tags are held onto the dugong using suction cups, so they're non-invasive. The suction's held on using a galvanic timed release, which is essentially a corrosive metal link. And depending on how hot the water is and how big that corrosive link is, determines how long it takes for the corrosive link to dissolve. Once that link dissolves, the suction releases, the tag floats to the surface of the water, and we spend a couple hours tracking down these tags using radio frequency. And then once I get the tag back, I can get the data. So to date, we've caught and tagged dugongs in 3 locations. So in Exmouth Gulf in Western Australia, Moreton Bay in Queensland, and New Caledonia. We've caught 30 dugongs. We've only got data back from 25. Unfortunately, some tags get lost, and it's a relatively new technology, so the tags will sometimes fail as well. But from these 25 dugongs, we've got more than 14,000 dives. That's a huge amount of dives. And 92% of these dives are in waters of less than 4 meters. And if you have a look in the corner, you can see a dugong in a stretcher that we've just put a mini diary on before it gets released. So what does the multi-sensor data look like? Well, it looks like a mess of squiggles at first. And when you spend hours looking at it, it makes a lot of sense. But it's essentially, if you were to keep a record in a journal of everything you did during the day, that's what we get back for these dugongs. So this is a 30-hour deployment for a dugong from New Caledonia, but we can then look at 1-hour segments. So at the top we have depth, and then we have activity, and then we have body angle, and then we have another measure of activity. But we can look at these segments, and then we can compare on a per dive basis. So if you remember back, quite a few slides back, I showed you the shapes of dives. There was one which was a U-shaped dive. Now, all the dives in blue would be classed as U-shaped dives. So we have depth at the top in blue, below that we have activity in red, and then below that we have fluke beat cycle, and that's presented as a spectrogram, which is essentially a heat map. So the warmer the colours, the more movement there is. This is all plotted against time. Now what I want you to remember is that the dive on the left is about 220 seconds. That's the same duration as the time from the start of the first dive on the right to the end of the second dive on the right. Now, if we look at activity, which is the red line, we can see on the left, there's not very much activity in that bottom phase of the dive, so that flat line at the bottom. And that's also reflected in the spectrogram, where it's cooler colours, so less activity. If you compare it to the dives on the right, there's considerably more activity during the bottom phase, and this is also seen in the heat map. Now, what's really cool about multi-sensor data is we get not just activity, we get the magnetometer data and we get the gyroscope data as well. When we combine this, we can recreate the dives in 3 dimensions. And at a quick glance, you can see although the dives look very similar in 2 dimension, they actually look very different in 3 dimension. Now, the 3-dimensional dives are coloured by elapsed time. So where red is the start of the dive and blue is the end of the dive. So on the left-hand side, we've got the dugong descends. There's a really little, I don't know if you can see, if you have really good eyesight, there's a really little stack of colour. So that just tells me that the dugong's spending most of its time, almost 200 seconds, at the deepest point of that dive before it ascends. Whereas on the right-hand side, the dugong descends, it then moves through the bottom phase of the dive and actually does a U-turn before it ascends to breathe and then descends again and moves through the bottom phase. So by incorporating activity data, we can actually reveal how the dugongs move through the water column, and it gives us a lot more information than just 2-dimensional dives. Now, this was all great, and this was fun to do and reproduce these dives in 3 dimensions. But then my supervisors and I were talking, and we were like, okay, if 92% of the dives is in less than 4 meters of water, how are they able to control their buoyancy in a way where they can dive efficiently? So dugongs, if you don't know the morphology of dugongs, they've got these big lungs that span the length of their torso. So very big. And buoyancy control is a very important influence on the energetic expenditure of an animal, in that a positively buoyant animal will need to actively swim to overcome positive buoyancy at the start of the dive. So there's a link between buoyancy, pressure, and effort. Now, if you're scuba divers, you'll know that buoyancy changes with depth as lung volume changes proportionally to the increase in pressure associated with the increase in depth. So at the deepest point of the dive, there's the most pressure acting on the lungs compared to the shallowest point of the dive. So buoyancy changes because of pressure. So what we can see in this plot is, at the start of a dive, if a dugong is positively buoyant, it will need to actively swim to overcome positive buoyancy. However, at a certain point in the descent, so there's the black line, there'll be enough pressure acting on the lungs that the buoyancy of the dugong can shift from positive to negative. The dugong can then glide the remainder of the dive. The opposite happens in the ascent. The dugong will need to actively swim to overcome negative buoyancy, but at a certain point, the lungs have expanded enough due to the reduction in pressure that the dugong can then glide the remainder of the way. Now, we can see this in the multi-sensor data. So at the top, we've got depth along the y-axis, and that's plotted in red. And then below that, we have acceleration or activity, plotted in blue. And these are plotted against time. Now, every peak and trough in the activity is a flute beat of a dugong. So we can see at the start of the dive, the dugong has to actively swim to overcome positive buoyancy. But around 6 or 7 meters, the flute beats stop, but the depth keeps decreasing. So dugong's gliding the remainder of the dive. And then the opposite happens as the dugong ascends. Now, if you remember, I had about 14,000 dives, so I had a lot of dives to go through and pull out all these numbers, and I did it manually, so it took a long time. But by getting each value that depth, that gliding depth begins in the descent and the ascent, I could find the relationship between maximum dive depth, and that's what we see here. So ignore the colours, we're just looking at the overall pattern. So we've got descent on the left and ascent on the right, and we've got that depth that gliding begins along the y-axis for both, and then maximum dive depth along the x-axis. So what we can see is there's a positive relationship between these two for both the descent and the ascent. Now, what's cool is that typically marine mammals will exhale before diving. So they have a relatively constant volume of air in their lungs when they dive. So this means that they would start gliding at the same depth, no matter the maximum depth of dive. Whereas shallow divers, such as turtles, are typically, well, not all turtles, some turtles, are typically inhalant divers. So they can control their lung volume based on the depth that they dive to. So dugongs are a shallow diving marine mammal, but they control their lung volume with dive depth, so diving more like a shallow diver, like green turtles or, I blanked on another turtle name, but anyway, compared to seals that will dive on exhalation. So now we've gone through a bit of physics. I then went back to my three-dimensional dives and started looking at the activity of dives to see what the dugongs are doing during their dives. And we have some examples here. So the top blue is acceleration and red is dive depth. And we can see the peak and troughs in the blue acceleration is the same as what we saw in a previous plot. So that's swimming, that's the flute beats of the dugong. In the plot below, in the bottom phase of the dive, so at the deepest point of the dive, we have lots of little up and downs, which is indicative of foraging. And when you're looking at 14,000 dives, you see these patterns a lot. So it's easy to say, okay, this is resting, this is swimming, this is foraging. But then when talking to my supervisors, they were like, well, that's great. How do you know that's actually what you're seeing? And since I wanted to work out the time activity budgets of dugongs, we kind of realized that we can identify patterns in the data of what we think is happening, but we had no way of validating the data. And this is where Malkut comes into my project. So Malkut provides a unique opportunity for us where we can put the tag on Malkut and then we can visually record his behavior. We can't do that with wild dugongs, so we put the tag on, off they go. They live in turbid waters. We don't see them again. So this is Malkut with his tag. He had his tag on Tuesday and Wednesday, and today. So while the tag's on Malkut, I'm taking visual observations for 30 minutes on, 30 minutes off. So we're writing everything that changes. So he might do 10 behaviors in the space of 20 seconds, or he might decide he wants to rest for 10 minutes. Either way, everything's recorded in those 30 minutes. Then what we're going to do, we're going to match the visual observations with the movement data. We're going to match them by time. We're then going to use the behavior patterns from Malkut's data to identify behaviors from wild dugongs. This will then enable me to create more accurate time activity budgets for the wild dugongs. So Malkut's behavior will help us understand the movements and habitat use of wild dugongs. Thank you. To close up.
Thank you so much. All right. Actually, we will continue with questions because I'm pretty sure you have plenty of questions. And I would like Jonathan Diaz from the YAS Research and Rescue Center to come on stage, and Dr. Das from the Environment Agency Abu Dhabi to join us on stage. If we can have some microphones here, and if you want Ronnie and Sarah to come back on stage. I'm going to introduce Dr. Das, but you should know him by now. Dr. Das has been studying the dugong, sea turtles, and dolphins at the Environment Agency Abu Dhabi for well over a decade. At the Environment Agency Abu Dhabi, he is involved with the assessment of critical coastal and marine habitats, and he is an active member of the IUCN Marine Turtle Specialist Group and regional chair of the IUCN Sirenian Specialist Group of Indian Ocean region. So he knows what he's talking about. And we have Jonathan as well. Jonathan Diaz Jonathan is currently serving as a senior manager of rescue at the center. In his role, he leads a team responsible for managing and coordinating rescue efforts. With over 20 years of experience working at SeaWorld's various locations, Jonathan brings a wealth of experience to his role. And I would like to first maybe have some images on the back and ask, I'm going to come and sit next to you, Jonathan, but first, can you explain how did you prepare Malkut for this week, and yeah, what was Malkut's role
Sure,
this week?
gladly. Honestly, before I get started, I kind of just want to talk about how excited I was that this project was coming. As Dr. Elise mentioned, been with SeaWorld for over 20 years. A lot of that has been with the rescue. And so it's always been very easy to talk about how rescue contributes to conservation, just because we are rehabilitating animals, returning them to a natural environment. We can easily tell that story. And then this project comes along, and it's very easy now for YAS SeaWorld Research and Rescue Center, as well as SeaWorld YAS Island Abu Dhabi, to kind of walk the walk, right? We talk about the great work that we do and how the animals that we have contribute to animals out in their natural environment. And now Malkut is the perfect example of that. So it was probably roughly a year ago when kind of these conversations started. And immediately just very excited. So first kind of learned about Sarah's part of the project with the morphometrics, and then Renee's part with the tag. And so even before we got to the point where we knew this was going to happen, we pretty much started preparing Malkut. And I can be up here for hours and not give my team enough credit. They did such a good job with Malkut, basically training as soon as we learned about this. So, oh look, visuals. So we had been taking measurements ever since Malkut arrived here. But then we started to kind of focus more on the measurements we would be using for this study. So by the time it was Malkut's turn to actually do some of these measurements, you can see a lot of these pictures are Malkut out of the water, but many of the measurements we took were voluntary, with Malkut in the water, the keepers and trainers handling him, and just moving down his body as he just sat there. And so our whole point, the whole point of what we were doing was to make sure he was nice and calm. This was just an everyday thing. And so we would take weekly, bi-weekly measurements, just another day at the job for Malkut. And then as we started getting closer to now realizing, oh, this is happening, and we are going to be putting a tag on Malkut, now it's time to get him ready to have something kind of attached to him. So we started very small. We started with one suction cup, and Malkut kind of took that very, very easily. You know, it didn't really bother him very much. We started with a very short time period, and then slowly would add time to that. When we added the second suction cup, he decided that he would kind of rather not have that for a spell. So he swam over, kind of knocked one off, and at that point we were like, okay, let's take a quick step back. What we didn't want is for him to rehearse that behavior, to know that if I go over to that rock and I just rub against that rock, I'm gonna pop this thing right off. So we shortened the duration that he had two suction cups on, and then continued to lengthen that over time, to the point where he was able to wear the diary tag not only during the day, but also for the past 2 nights. And Renee could probably talk to this a little bit more, but she had talked about in our conversations kind of information bias, where if we were only getting data during the day, that doesn't really take into account what he's doing overnight. And so with that information in mind, we decided, hey, let's get him to the point where he can wear this overnight. We got 2 good nights of data, and really hope that you were able to get a lot of good information from that. So that's kind of the process that we went through. It's really the same type of training that we do all throughout the park with all of our animals. We just applied those principles to Malkut and went from there.
Amazing. Dr. Das, do you want to add anything before we take the first question?
In fact, in Abu Dhabi, we tried satellite tagging of dugongs to just find out their migration pattern and habitat utilization and where do they go and all this. And we got everything from Australia, from Darwin Coastal Engineering Department made one teether for us. It was in 2008 and 2012. I don't know how they caught 25 dugongs. To catch 2 dugongs, we spent 8 days. One helicopter locating a dugong, one large boat chasing the dugong, a small barracuda boat catching the dugong using rodeo method. Rodeo method is that you tire this dugong out and jump over it and put the day loop in the tail and then fix it. Did you use rodeo method to catch?
Yeah, we did rodeo.
Okay, there are dugong catchers in Australia, we can get them. Now, coming back, we tried the teether efficiency in Australian water, the biggest mistake we did. Because Australian water with standard sea temperature, sea water temperature and salinity didn't work here. They tried and they said it will go for 6 months. So we were happy and we were waiting for, after all these logistical preparation, expenditure and all these, we thought we can get really valuable data. What happened? None of the teethers stayed for more than 3 weeks, just 21 days. And probably our dugongs are lazy enough, 7 kilometers per day movement, and only one dugong moved 32 kilometers from Butina to Al-Bajim. So now the question is, it's a wonderful thing what Sarah is doing, that methodology of body mass index and all these things can give us. We have drone now we are using for behavior study, we can use for this once their methodology is established. And what Renee has done is again wonderful, and we can use these small suction cups and all this. But how to catch 20, 25 dugongs, that will be the biggest challenge for us. I think SeaWorld will come as the great help for this.
We'll be happy to
But
help.
you know, here our management and leadership is very sensitive to look after the welfare of the animal. Any stress, anything, we'll have to abandon this one. Now, coming back, the research question, if you are doing this study, for both of you this question, with a dugong that is well fed, nicely using with, means water temperature not changing, no climate change of like no temperature, the standard depth and all these things, are you going to have problem when you apply your logarithm in wild animals, wild dugongs? And what about, this is a subadult. Now, if you are going for adult dugong or calves and all these things, how much standard error you are going to get?
I can go
Sarah,
first.
you wanted to start?
Oh, I can use this.
Yeah, you can use
It's right. Sorry again, the comparison was with a well-fed dugong like Malkuth or?
Malkuth, yeah.
About the error, well, there are, I suppose, several things to consider for photogrammetry terms, because Malkuth is a juvenile dugong. He is not an adult yet. So we are still getting like the body shape representative of a dugong, but of a juvenile dugong. And therefore, we will have a good reference for juvenile wild dugongs. But as many animals, when we grow, we might change a bit of our body shape. For example, babies have bigger head relationship with their body than adults. It might be the same with dugongs. So there might be a slight error to apply the body shape of a juvenile for images of adults. But there are only 3 dugongs under human care in the world. So even having this information is very, very helpful for us. Yes.
It will be very helpful for us also. Your result will actually help us to design more studies in this region. Thank you very much.
Thank you.
Thank you. We'll take the first question. All right, Mohamed.
So before I ask my question, I'm going to cheat a bit, and I'm going to ask 2 questions.
All right, 2 questions.
So earlier in your presentation, where you showed the dugong maps, their diving maps, you told that you couldn't figure out what they were doing in the time and their depth. So why can't you just put a camera on them?
Yes,
Yeah.
I would love to. They're very expensive. So they do exist. They're about $20,000 a tag. So my project was not scrappy, but we had to work hard to get funding to get the tags that we got. So we ended up with 4 mini diary tags. The one camera tag we did deploy was lent to us by Parks and Wildlife in Western Australia. But they run projects on flatback turtles and green turtles, so couldn't lend it to us more than the time that we borrowed it. But yes, if you have a well-funded project that runs, has lots of money, can afford camera tags, that is the ideal situation to be able to validate behaviors.
So my second question is, both of you were using Malkuth to prove just to observe him. So he was raised in captivity. Wouldn't his behavior be different from the ones in the wild?
That's a good question. Yeah.
As for you, yeah?
Yeah, so probably one of the biggest differences I've seen when I've just quickly looked at the data is that Malkuth does a lot of surface resting. And a dugong in the wild, hardly any of my dugongs do that. The ones in Exmouth, it's quite a shallow water gulf. So the ones in Exmouth do shallow water resting, presumably because there's less risk of sharks if they're resting in shallow waters. Whereas in New Caledonia and in Moreton Bay, all the dugongs there did deep water resting. Bottom resting in deep waters, because if you're shallow resting in deep waters, there's an ambush risk of a shark coming up to get you, basically. Yes, the other difference could be the foraging, the feeding signal. because malkoot's feeding on lettuce. I can't remember what type of lettuce. Dugongs in the wild are cropping or excavating seagrasses, which are quite little. So it could be a big movement difference. But with those, surface resting is the body's still, and that's the same no matter what depth you're at. If you're resting, the body's still. So the acceleration pattern and that activity, still there'll be no activity if you're resting, whether it's at the surface or at the bottom. Foraging, it's still an up-down movement. I have to compare a little bit between the two because this is fresh data. I'm hoping it's not too big a difference, and there's enough difference between signal of a dugong swimming, of a dugong resting, and dugong foraging to be able to separate those three behaviours.
Thanks, brilliant. Thanks. I have another question over there. Do we have another? All right, I'm coming with my microphone. Oh.
Yeah, thank you. My question is on the part of photogeometry. Do I get the word right? Because I understand objects in water appear smaller than they look on the ground. So how do you make sure the data you collect from the guys, the length and the width and all that, how do you make sure the data is accurate? Because in water, they look smaller than they actually appear. Thank you.
Yes, yeah, that's also a good question. Actually, most of the images we are using are from dugongs that are surfacing for breath. And so therefore, there shouldn't be a lot of distortion due to water because they are very close to the surface or in the surface. There will be probably some degree of variation because there are still part of the body that is still under, but it shouldn't be much. Also, we are looking at the relationships in the body, so length and width. And when we work with ratios of measurements, the ratio or the error should be maintained in the ratio. So at the end, you kind of annulate that. Does it make sense? Yeah, thank you.
Great. We have another question over there. Yes.
Hi, my question is for Sarah. Thank you. Both your presentations were amazing. The first is, do you extract your images from the video or do you take still photos? You extract from the video?
Yeah, from the video.
Okay, and my second question is, how do you distinguish between individuals to make sure that you don't repeat measurements?
Yeah, that's a good question as well. Dugongs don't have very distinctive marks as humpback whales or other animals. So it might be very difficult to distinguish one individual from another. However, we have noticed that when we find a big group, we have also worked our methodology in a way that we start from the individuals that are on the, let's say if it's a group feeding, we will start from one side and go like this, and then trying to don't overlap or cover the same individuals. There might still be some degree of risk that we are repeating individuals, but we have also noticed that some dugongs have very shallow marks on their backs. Dugongs have tusks, and sometimes when they interact, they get some scratches. So these marks are very short term, but from videos of the same day or very consecutive days, it is possible to identify some individuals from others. So yes, those little marks have been helping with that. Thank you.
Nice. I guess, all right. Yeah. If we can go this side. Yeah, yes. Thanks, Dr.
Hi. Do dugongs, could they survive by themselves?
So you mean when they're young?
Yeah.
Yeah. So do you want to answer that?
Sure. You'll see large gatherings of dugongs, right? But a lot of times in those large gatherings, there's mother-calf pairs, But then as dugongs start to get older, they're just going to be moving towards where those feeding grounds are. So you may have several solo dugongs in the same area, but they're doing just fine on their own, because they're just finding those feeding grounds. So they may be surrounded by other dugongs, or they may just be off on their own for long periods of time where they're just feeding.
Okay, thank you.
All right, yes.
In the measurements that
Yeah.
dugongs that you use, that you captured and release again, is it possible that you considered using a calibrated tank where you immerse them in water and then use that method to measure their body volume?
Sorry, again, the last part of the question, please.
Have you possibly considered using a calibration tank where you take the level of the water with the dugong out the water and then submersing
okay.
and then
With the
actually
dugong
measuring
captures.
the volume? Yeah.
Yeah, I would say that that is very difficult because the dugongs that are captured, they're just held for maybe 10 minutes at most. It's part of the protocol to not stress the animals. They are captured, measured, tagged, and released. It would be very interesting, but yeah, it's not possible to do.
Yeah, it was just a thought. Thank you.
Thank you.
Thank you. Yeah, Mark.
Yeah, thank you for great talks. I had a question about dugongs in Australia when you showed the map, because I know traditionally dugongs were hunted by indigenous people, especially in Cape York Peninsula, Torres Straits. And I was surprised that none of your data was from that area. Is there a prohibition to collect data in that area? Is there some degree of protection or something? Because obviously there's indigenous knowledge. The people in that area are very good at catching dugongs. I was just surprised that you didn't have any data points in that area, because I know that there's a lot of anthropology literature about that area and about traditional use of dugongs. I just thought it's a shame that there was nothing from that area when it's kind of, as I understand it, the greatest concentration of dugongs in that area in terms of population.
For my work, running a dugong catching field trip is quite expensive when you're close to a major city anyway. So when we were working out where we were going to catch the dugongs, initially Cleveland Bay, which is where Sarah works out of, was one of my sites. But the water's very turbid, so we had to weigh up the cost of catching. In some areas where dugongs are hunted, they know the sounds of boats. So when they hear a boat, they disappear and you won't catch a dugong. And when we were talking with my supervisor, Helene Marsh, that's the case in Torres Strait. So often, from what I understand, and this could be a little bit wrong, we'll see, is that in Torres Strait, they typically, Indigenous people will wait on platforms, and that's how they catch dugongs. Which, if we had to rodeo a dugong doing that method, that would be very difficult, because you'd have, no way to chase them to tire out, and your catchers would get beat up. They have started a program in Broome, with working with the Indigenous rangers in Broome, catching dugongs and tracking them there. So far, that's just GPS. But I think for the most part, it's, cost, access, the chance of you being able to catch the dugongs if it's turbid. Yeah. Mm. Yeah. Yeah. Yeah, that's a good idea. Yeah, I guess in Australia we have crocodiles often overlap with where dugongs live. And I know when I was trying to get permits to catch in certain places, James Cook University was very aware that there are crocodiles.
Yes, that's another issue. I guess, yes, here for sure, next one. You have mine. Yes.
What are the risks of deep diving of these species, and what are the adaptations to prevent it?
So do you mean, I might just need a bit of clarification. You mean the risks like physiologically?
Yeah.
Yeah, so I can, well, I guess if they're having to dive deeper, having to work against more pressure, so it'd be more energetically costly. The deepest dive recorded for a dugong is 36 metres. So in the grand scheme of how deep some animals dive, like some seals dive deeper than 1,000 metres, they don't have the risk of like decompression sickness or barotrauma, so they don't have those risks. But yeah, I guess off the top of my head, the main one would be the cost. Yeah.
In fact, it depends on what is your definition of deep. Dugongs live in and around seagrass areas. So it depends how deep your seagrasses grow. That is why before doing dugong survey, we always do seagrass mapping. And in that, for example, Abu Dhabi waters, we have got seagrasses, good seagrasses, enough for a large herd of dugong to graze, are within 12 meters of depth because our turbidity is high. Water quality is not like Australia where you can see even 40 meters down there. So in our case, now slowly, probably due to climate issues and other things, our seagrasses are now reported from 17 meters, 16 to 17 meters. They are going deeper. But they are so scanty that not sufficient for dugongs to graze. And dugongs have to surface in 6 to 10 minutes, 6 minutes to 10 minutes to breathe. So that way, and buoyancy, what you heard they were presenting, positive buoyancy, healthy body and all this. So 10 to 15 meters in Abu Dhabi waters, enough for them. They don't go deeper than that. And they're shallow water animals. So it is up to seagrass and your, extent of growth.
It's all about seagrass. Over there.
Hi guys, thanks so much for the talk. I guess I'm wondering where the dugong is in its, which are its predators? And then further to that, you said that they live in herds. Do we know how they communicate with each other? Do they use sonar or something like that?
I would say in, Abu Dhabi waters, we have seen instances of predation from orcas that would occasionally come in, feast and then leave. I'm not too familiar with threats in Australia.
Australia is sharks and crocodiles.
And sorry, what was the second part?
How do they communicate?
So from what we've seen, they communicate with very high-pitched squeaks. And so it's you'd think a big animal and you think like an elephant and the relation there is this deep baritone, but really even adults have a very high-pitched squeak or whistle. And so a lot of times you'll see the front part of their muzzle just kind of move here and there. That's kind of an indication that they're vocalizing.
And Jonathan, you can mention also when dugong calves are rescued, adults and calves have different noise level, the way they communicate. We have not studied. Frankly, there is no data or there is no study from this region at least. We are now using drones to study the mother and calf baby behavior. But when we see calves abandoned or washed ashore, still live, they have a cry kind of thing like child, human, this one. And very, very sad and pathetic noise comes from them. And that is what Malkuth, when Malkuth was rescued, how many years? 6 years back? Yeah, 6 years back. From Marawa Marine Biosphere Reserve, he was holding one pole that in the jetty and like holding mother and then struggling there. And there's a cry kind of thing, not whistling sound which adults give. So that is not the time to record these or to keep data. Our main objective is to rescue this animal, take it dehydrated, and all those things. But there are different things. No, not much study done.
But is it? Do you know if there any study or proper research on that communication or the vocalization of dugong? Is it something that Helen has been working on, maybe, or any?
Helene's not recently. There has been work out of Thailand. We did think about it for my PhD because the tags, one of the sensors you can add it is a hydrophone, which would have recorded dugong sound, but came down to cost of the tag. So yeah.
There are lots of studies on cetacean communication, and we are using PAM to do that. But for Sirenians,
yeah, exactly.
It's
not much. Huh? Okay, something to do then. All right. I had one question here, and then we'll take one more over there. Oh, yes. And that will be the last.
How much seagrass do dugongs require on a daily basis, considering how much energy they use?
From the literature I've read, it's about 10% of their body weight. So an adult can weigh like 300 to 450 kilograms. So they need about 30 to 45 kilograms of seagrass a day. Huge amounts.
Yes. Oh, wow, thank you.
All right. Yes, over there. Yeah.
Thank you very much for sharing your research with us and choosing us to be a part of it. This question is actually for everybody. I thought it was very interesting when you said that our seagrasses here in Abu Dhabi are actually going to deeper. And so with that, do we know what they're capable of with using the tag? Like how deep are they able to go? And with the retreating seagrasses going deeper, do we think that there's a concern that food availability is going to be harder for them?
Yes.
So the deepest feeding dive I have recorded, or what I think is a feeding dive, yet to be validated, is 12 meters. But off the coast of Indonesia, they've spotted feeding trails at 30 meters. So it probably comes down to, well, at the moment there are shallow seagrasses available, so it's easier for the dugongs to access. I think as the shallow seagrasses disappear, they're going to obviously be forced to either move somewhere else that has shallow seagrasses or adapt and dive deeper. Now, how they sustain that will probably depend on how much seagrass is in the patch, the quality of seagrass. So if the quality of seagrass is higher, they may not need to forage for as long, but yeah.
I think you need to know here something to tell you about biology of dugongs. Dugongs are not great swimmers, first thing. Look at their body, round, rotundus and all these. Small flippers. And, peduncle is this one. It's not the animal that can actually swim, dive deep water. That is why they are called coastal animals, and they stay in and around seagrass areas, 12 meters, 13 meters, like that. And during very high sea, cyclone, usually dugong and their mother and calves will take shelter when shamal comes in Abu Dhabi, Arabian Gulf, I'm saying. Shamal means northwestern strong wind. They will go to southeast side of the island and take shelter. We have several studies in when shamal for 2-3 days, mother and calf will get restricted because those high winds, waves also, they cannot surface and breathe easily. So they are not great swimmers, neither they can swim deep sea or face cyclonic weather. They are not like dolphins. So that is where the problem comes, and they always remain in those areas. And I think Australia, there are some old reports, after cyclone, they have seen some dugongs with lots of injuries in cliffs and rocky shorelines and all this. So those type of, so they don't go to deeper water, usually, don't prefer. If there is, dugongs move for two things, food and reproduction. If both available in shallow water, they will be there in shallow water.
All right. I think we're going to close here. I'm sure there would be a much more question to ask, but thank you so much for coming all the way to see us and to see Malkut. Renée, Sarah, and thank you so much for sharing your research with us tonight. Thank you, Dr. Das, for being here tonight as well, and Jonathan, obviously. Thank you for coming. I hope you enjoy the talk and learn more about the dugongs, the seagrass, and our beloved Malkut, obviously. Next talk, keep an eye on our website. We don't have the confirmation yet, but we should see you very soon again. Thank you very much. Thank you.